on for a second.
There you go.
Okay, good. Okay, can you see the pointer?
Where'd it go? I thought
It's
it's
around here.
No.
Yeah. Okay, good. Okay, so thank you very much for organizing this and the opportunity to see this exciting new data. As you know, we started working in CA2 time ago, and we were really intrigued by this region. The motivation actually was trying to integrate CA2 into the concepts of how the hippocampus function is implemented. So we know how the different regions from the entorhinal cortex represent different aspects of memory, or the items that we need to get to memorize. And in the episodic memory function of the hippocampus, we have this conceptualization of that the what, the when, and the where somehow are represented that the tuning curve of single cells, which then project specifically across the different regions in the hippocampus, and from the entorhinal cortex by the classical trisynaptic circuit into the dentate gyrus. And here there is functions regarding the pattern of separation properties, so that the representation could disambiguate between contexts from the dentate to the CA3, where pattern compilation could be implemented, and of course in CA1, where many representation could be integrated. I mean, on top of this, there was emerging data suggested that there is a weird region here that we keep weird, as Serena suggested, in CA2, which can actually represent how time could be evolved, and how across a particular exploration in a room which is fixed, but there are some temporal signals that help to understand how this is being represented. And also the fact that we can represent aspect of the who, I mean, social information and how we relate with another. So this is more or less the motivation for me. And also because, as you probably know, we are very much interested in bringing the complexity of the cell type specificity and the different subcircuits that wires the entorhinal cortex and the hippocampus, and across hippocampal regions and within the different hippocampal regions. And you probably know the data from my lab regarding the deep and the superficial CA1 cells. And also we were intrigued of how this CA2 region is connected with the CA deep and superficial pyramidal cells in CA1, and how the different events that are generated in hippocampus emerge according to the activity of these different cellular populations. So the point was trying to understand CA2. And we try to do this in vivo, but this is a very hard task because, as you probably know, the CA2 is a very tiny region, and we were trying to target the region in head-fixed animals, head-fixed rats running in a wheel. And so we had to learn how to identify the border between CA1 and CA2 using LFP signals. And there were some indications at the beginning that were guiding us that emerged from the observation in Bushagi's lab regarding how sharp-wave ripples could be the shape of sharp-wave ripple events around the CA2 are relatively distorted. They are typically running just before the sharp wave when you record them just in the border. But we were also started noticing differences in how theta activity could be detected there. So when you have your electrode going through CA2, you can see how theta is strongly attenuated. So if you have a reference electrode in another region of the hippocampus, like for instance here, we have an electrode in CA3A, and we have an electrode going through the border with CA2. So you see that there is a difference of the amplitude of theta that indicates that you are around the region. This is another example of another penetration going precisely through the CA2 region. So as we said, if we align the sharp-wave ripples recorded across a different region by the peak in the CA1 the event, you see that in the region of the CA2 there is this earlier contribution of the high frequency oscillations before the peak. But this is also happening in CA3A and B and C, and was not that easy to identify. When we look at the expression of the power of the different oscillatory bands, like theta or gamma, around the border, and we define the border here, as you will see later, at the tip of the MOSI fibers, we noted that there were some heterogeneities, some inhomogeneities in the area that was characteristic of the CA2 region. These are for electrophysiological labs who wants to target the region precisely, can follow this tip to try to identify where they are blindly when they penetrate such a small region. Now, how to define CA2? When we started working, we were working in rats, and for us it was very clear that the tip of the MOSI fiber were not terminated where we saw PCP4 positive cells. That means that CA2 in the rat somehow could be differentiated into a more proximal region and a more distal region. And the distal region was not receiving the inputs from the dentate gyrus, whereas the proximal had. We also noted that some PCP4 cells in the rat were located in the deep locations in the CA3 area. And we suspected always that these cells here are actually CA2 cells, which are probably, because of how you make the cut, penetrating in this direction, but also it could be some heterogeneity in the organization of the CA3 at the distal regions. But then when you confronted this with the mouse, as you see in the mouse, most of the CA2 region in mouse is innervated by the proximal. The proximal CA2 region in mouse is innervated by the dentate gyrus, and the tip of the MOSI fibers can be defined here, and very few PCP4 cells are located distally from there. So we started suspecting that there were some differences between species. Another marker of CA2 that works well with immunostaining is alpha-actinin, and we use that one together with a good marker of dorsal CA1 cell, which is FS1, which allow us to identify clearly the shadow of the MOSI fibers, the position of the CA1 cells, and expression of CA2 cells. So using alpha-actinin and PCP4, we look for the distribution of alpha-actinin positive PCP4 positive cells, alpha-actinin negative PCP4 cells, and alpha-actinin negative cells, FS positive cells, which are presumably the different types. So these are CA2 cells, these will be CA3 cells, those will be CA1 cells. And if you look at the distribution of these cells around the border, we call CA2 proximal, distal, and CA1 and CA3, we clearly see that there is a different expression level here, that there are an intermixture of population within the CA2 area. So within CA2, there are some few CA1 cells in the rat, which actually are more, the density is larger in the distal as compared to the proximal. And in the proximal, there is an intermix with CA3 cells, and then PCP4 positive cells, which are around the area. So the region was strongly heterogeneous in terms of cell types from CA3, CA2, and CA1. So to try to clarify this better, we implemented single intracellular recordings in vivo in anesthetized rat. So we impel with glass pipette individual cells that we stain with neurobiotine for posterior identification regarding the presence of thorny excrescence, which were characteristic of C3 cells, cells which do not have thorny excrescences, which at this time were considered PCP4 positive and therefore CA2 cells, and cells expressing the FS1, which were CA1 cells. These data are the first, and as far as I know, the unique data that is available today on single cell recorded intracellularly in vivo in the rats. And this is unique because we need to inform the models regarding how the physiology of these cells are. The interesting thing is that what we noted with these single cell approaches in vivo was that the expression of theta modulation was strongly different for CA3 cells, C1 cells, and within the proximal and distal CA1 region. So this is phase locked firing of the different cell types. As you can see, you can note the difference organization along the theta oscillations, so that the modulation is stronger for the distal PCP4 positive cells as compared with the proximal, and there were differences in gamma modulation across the region. And we team up with Azara, who examined her data according to the proximodistal location in freely moving animals, because these data were essentially from anesthetized animals and some from juxtacellular recording head-fixed animals. But to go massively, we asked her to examine her data, and she noted in freely moving rats that this shift in phase and the increase in modulation was present in freely moving animals. So the other thing that was extremely interesting in functional terms was the fact that depending on the inputs, responses were differences. So these are excitatory potential amplitude for the different cells in response to C3 inputs, stratum lacunosum moleculari inputs, and inputs from the dentate gyrus. We specifically activate these different inputs in slices. And as you can see, there is clear trends, proximodistal trends, which clearly identify the different cell types. So the red ones are CA2 cells, confirmed by PCP4. The green ones are CA3. The blue one are CA1. Those cells which are not marked could not be identified. The bottom line here is that the proximal region of CA2 is receiving input from the dentate gyrus and the CA3, and they are modulated differently, poorly during theta, or strongly during gamma. Well, however, the distal region of CA2 in the rats receive more input from the entorhinal cortex. They do not receive input, obviously, from the dentate gyrus, and they were lower in gamma modulation. So these functional differences prompted us to look a bit more and to be interested in CA2. And we wonder whether the differences are functional differences that were reporting in the literature were expressing this heterogeneity. So this is, we heard Serena about this beautiful and interesting data would actually point to the fact that, as we were discussing in this session, CA2 cell could be actually heterogeneous itself. There could be different expression of the different markers. And this, as we will see, will be very important across species. If you make slices, horizontal slices, and this connects with what Michael was saying before, and it is important for in vitro studies. If you make horizontal slices and you stain these slices from PCP4, you will see how the PCP4 positive cell distribute all along. So some of the conclusions that we are reaching in vitro should consider also the identity of the cell types, because as you can see clearly here, there is a deep layer of PCP4 positive cell extending through all along CA3, the regions CA1, and into the CA2. And more importantly, if we make a coronal section from the dorsal to the ventral hippocampus in rat and in mouse, we can clearly see, as again was something commented this morning, how the CA2 positive PCP4 cells, PCP4 positive cells could be also identified at the border, an intermediate level. And this recall pretty much some of the data suggesting that deep CA1 cell could be involved in social function. So we think that it is important to clearly identify the cell types and the conclusion that are energetic could be attributed to the different regions, and this heterogeneity in the configuration of the circuit should be considered. Now, as I said, something that was clearly striking for us was that fact that there were clear differences in the CA2 organization in mouse and rats. And also others has been noted this. We were looking at the fantastic special issue of the Hippocampus regarding CA2 and indications of differences across species that may require some specific staining so that we can clearly conclude where and what are the limits of CA2 as we were discussing. And we hear about this, the recent work by Serena in foxes, where using Cal binding could be identified that in this case, for instance, the proximal regions of CA2 is actually a bit smaller as compared with rats and mouse. And in this particular section, we saw a clear expression of Cal binding in CA1, where we know that Cal binding is expressed in a subpopulation of cells, which in rats and mouse adopt a deep location. We started collaborating with other researchers, and we were trying to dig into this for time ago. We have been trying to understand this closely. And this is an example from ferrets. And we got the sections from Victor Borrell in Alicante, in Spain. He is a specialist in looking at the ferret brain. And here too, as well as in the fox, Cal binding is expressed in the gyrus, and of course you can map the mossy fiber, but also you have expression in CA1 that doesn't allow you to clearly identify the region. But somehow we need to look at other species to try to identify how this region is organized. So one of the collaborations that we started was with Carmen Cavada here in Madrid from the Universidad Autónoma. And Carmen has unique material from human who were, she collected this over the years using very careful perfusion strategies to obtain very high quality immunostaining analysis. And when we started playing with some of this material, we noted that PCP4 is not working well in human. As you can see clearly here, in human, PCP4 is staining the mossy fiber clearly, but just after, and this will be the definition by Laurent Denot, the region just after CA2, which is corresponding to CA2, is not expressing PCP4. If you look at chromogranin, which is a marker for CA2 that was previously reported, you clearly see how you can stain these cells specifically as compared with PCP4. And actually the mossy fibers, the limit with the mossy fiber, which is just here, that form this kind of shape here, is clearly identified, and chromogranin positive cells could be also identified here. And in human, what we noted was that RGS14 is actually expressed in CA2, and you can clearly identify the limit of the mossy fibers, and you can look for the expression of these RGS14 positive cells here in the limit with the mossy fiber staining the CA2 region. So we decided using RGS14 to examine mouse, rats, and human, and monkeys. And here we team up with Ricardo Insausti, who will be talking just after me, and sections from monkey that I'm showing here and from humans are also collected from Ricardo, who also have been collected this material from human with very careful perfusion strategies to preserve the immunoreactivity. And he also have collected this material and have got serial sections in an ordered way so that we can cover the entire hippocampal regions. And in here, as you can see, we can use cal binding to identify the limit of the mossy fibers. We can use LGS4 to identify CA2. And I think it's quite clear that the evolution from the proximal distal organization is having a strong differences across species. So for the mouse, it's larger, the proximal as compared with distal, which is something that we can appreciate in rats, and that the distal portion is a bit larger in rat as compared with the mouse. And these become larger in monkey and strongly larger as compared to human. These are proportions of the proximal and the distal CA2 region defined along the cornus harmonic linear aspect. And as you can see, there is an increase and a shift in the proportion of the distal as compared with the proximal. And there is also a change in the proportion of CA2 in this species. And this is occurring at the expenses of CA3, not of CA1, because the percentage of the cornus harmonic, which reflects CA1 regions across the species, is relatively conservative. So the definition of Lorente said that the limit is at the mossy fibers, and also that the size of the soma decreases are the CA1. So we also measure the density of cells in monkeys and the area of the nucleus in monkeys. And we clearly see that using LGS14 allow for the definition of the proximal and the distal CA2, and that this is consistent with Lorentz definitions, which of course is not clear in other species. And this is for humans, similar as for monkey, the density of cells across the regions and the area of the nucleus. So CA2 in human, as well as in other species, is functionally organized in proximal and distal regions, and the inputs converging in the area are important to determine the functions. This proximal CA2 is innervated by the dentate gyros, and the distal are receiving more strongly input from the entorhinal cortex. This is something also that we are trying to confirm in collaboration with Ricardo, and I think he will probably show part of this data. And just to finish, a couple of slides to remind that the hippocampus, the human hippocampus, is way more complex than the rodent hippocampus. The human hippocampus has a head, which is very convoluted and very difficult to understand. Here is a section at the level of the head, and this is the work done in collaboration with Carmen Cavada. As you can see in the head, here staining for RGF14, that you can see, I'm sorry, for chromogranin, you can see the location of the CA2 at this level. And what I have been showing is essentially at the level of the middle hippocampus, which is comparable with the body, is comparable with what we have seen typically in rodents. And if you move to the tail, organization there is even more complex. And typically, what is going on in the tail and what is going on in the head is something somehow neglected. But I have to mention that for the electrophysiological recordings in human, because those are guided by clinical criteria and mostly target temporal lobe epileptic patients, most of the recordings are typically going through the anterior hippocampus, and this is coming through close to the head. And therefore, interpretation of how the signals and the local free potential signatures of theta, gamma oscillation, as I was commented at the beginning of my talk, were strongly reduced in the area, it could be also affecting the conclusion that we may be reaching if we are not considering the structure of the region. So to finish, I hope, some of these data from my lab could help to shed light on the functional organization of CA2, which is far more complex than we originally thought, that we believe, and I think that this is something that the community is also appreciating, that there is a proximodistal organization of the structure of CA2 and function. And this is very, very important across species because the molecular identity of the CA2 region and the organization is very variable. And we think that this has a strong implication, the structure and function of CA2, to understand their role in memory process involved in the hippocampal function. So with this, I just thank you, and I would like to thank people in my lab and of course the funding agency, but most of the work that in CA2 is carried on by Elena and Beatriz, and I have to acknowledge and to appreciate and celebrate collaboration with Ricardo and with Carmen, who has given us the opportunity to open to the human CA2 and monkey CA2, which is an amazing region that hopefully can help and shed light to the real function. So thank you.